Hi all, welcome to NEET Biology classes of Brilliant Qatar. Now I think all are confused regarding how to score better in biology for the NEET exam in a short time duration. Whether it is possible and all, there is a small confusion in the mind of students. Yes, it's possible. How it is possible also, let me tell. You have to keep a short strategy of how to practice for the NEET exam. This strategy you have to follow from the beginning till the end. Biology scoring is simple if you keep this strategy in your mind. The strategy is like this. Now let me explain. You have 100 questions from biology part in the NEET exam. 50 will be from botany and 50 will be coming from zoology. In that, you have to attend only 45 from botany part and 45 from zoology part. So, you know that the total number of chapters that is coming in the biology is only 32. And the number of questions that you are getting is 100. In that, you should attempt only 90 questions. So, from the 32 chapters, 100 questions are coming means maximum number of questions that you can expect from a chapter is coming around 3 or 4. Some chapters it will be less than 3 also, 2 or 1 question also will be coming. So, how you can increase the possibility or probability of winning the questions from each and every chapter? The strategy is simple. That is, what you should do is practice 200 questions per chapter. So in a short duration, is it possible? That will be going in the mind. Yes, it's possible. What you can do is, first you have to attend the Brilliant Qatar's question discussion. We will be doing 100 questions from each chapter. That will cover NCERT line by line. Each and every line of NCERT I'll be covering in the discussion of biology. So after attending this correct 100 question in the NCERT order, you can have the revision of the chapter also. After this 100 questions, what you should do is practice 100 question from our material. So after practicing 100 questions from our material, you have completed your target of 200 question, revision of the chapter and practice of the question. Then write a mock exam of 25 to 30 question from per chapter. So now you have reached a target of 230 question, from which only the same questions may be coming for the NEET exam. Chances of repetition will be very high. So the possibility of winning question is also very high when you do it like that. So same strategy you can follow for each and every chapter. So 32 chapter, 100 question. Very simple. You can get it easily from the Brilliant Qatar's classes. Okay, then let me give a capsule of 100 question from a chapter going through each and every line of NCERT. Let me start with the first discussion, Sexual Reproduction in Flowering Plants. This chapter is having a good weightage in the NEET exam. So I'll be discussing some questions from this chapter. So we will start discussing each and every lines of NCERT along with the question and some concepts also. Hope you will enjoy the session. Let me start with the questions. First question. The end product of sexual reproduction is, in angiosperm, the end product of sexual reproduction after double fertilization is, you can see the ovule will be turning into the seed and the ovary in the flower will be turning into the fruit. So here the fruit and seeds are the end products of sexual reproduction. So here the end products of sexual reproduction is fruit as well as seed. The fruit is formed from the ovary and the seed is formed from the ovule. Hope you got it. Next question. Floriculture deals with. Floriculture is actually dealing with the flowers only. So here, next question. Find A, B, C and D. So the diagram of a flower that has been given in NCERT that we would be discussing. So here, A is actually representing, in the given diagram, A actually represent the filament. And B will be representing the style, and C will be representing, C here that is given here, it will be representing the ovary, and D will be representing the stigma. So this is the structure of a typical flower that is given in NCERT. So we will just discuss the structure of a typical flower. A flower will be attached to the stem or branch with the help of a stalk-like portion. That stalk-like portion is called as pedicel. And the base portion of the flower that is actually called as thalamus or receptacle. You should understand on the thalamus or receptacle there is nodes and internodes. The nodes will be having modification of leaf. First node you can see the leaves are modified into the green color photosynthetic part of the plant that is actually protecting the flower in bud stage. It is called as calyx. Single units are called sepals. Main function is protection of the flower in bud stage. Protection in bud stage, that is the main function. Then next node you can see there is the beautiful and colorful part of the flower that will be attracting the pollinating agents, that is corolla. That is corolla, and single units are actually called as petals. Main function is attraction of pollinating agents. Attraction of pollinating agents, that is the main function. Then inner to that only the male reproductive part of the flower will be coming. This is the male reproductive part of the flower that is called androecium. And single units are actually called as stamen. You know the stamen will be having a thread-like portion filament. At the tip anther also will be coming. Then at the center is the female reproductive part of the flower. The female reproductive part of the flower is gynoecium. The female reproductive part of the flower is called gynoecium. Single units are called pistil or carpel. Single units are called pistil or carpel. It will be having a swollen bottom, elongated style, and at the tip there is a stigma also that is coming. Anyway, these two are involved in sexual reproduction in the plant. They are called as essential whorls of a flower. They are called as essential whorls of a flower. Whereas calyx and corolla, it's not involved in sexual reproduction. Hence, they are called non-essential whorls or accessory organs in a flower. Now let me go to the next question. Choose incorrect statement. Several hormonal and structural changes are initiated which lead to redifferentiation and further development of floral primordium. So differentiation, de-differentiation, redifferentiation if you are taking. Redifferentiation is a method of formation of permanent tissue from secondary meristem. Actually the flower is not formed from the secondary meristem and all. So the first statement that is given is incorrect. Then inflorescences are formed which bear the floral buds and then the flower. So inflorescence you can see there is a group of flowers that are arranged. So here the second statement is correct. In the flower, male and female reproductive structures, the androecium and the gynoecium differentiate and develop. So the formation of stamen as well as the pistil, that is by differentiation only from the meristematic tissue. That is correct from the floral primordium. So here A statement is incorrect. Next one, reproductive organs of a flower does not comprise androecium, stamen or the gynoecium. All these are reproductive part. Androecium and stamen is the male reproductive part. Gynoecium is the female reproductive part. But tepals are actually not coming under this. So what is tepals? In families like Liliaceae, you might have learned about morphology of flowering plants. In Liliaceae family there is no differentiated calyx and corolla. Undifferentiated calyx and corolla is coming. So that undifferentiated sepal and petal is called tepal. There you can see the sepal as well as the petal is fusing together to form a single structure that is called tepal. Group of tepals are actually called as perianth. So what is perianth? Undifferentiated calyx and corolla is called as perianth. In that undifferentiated sepal and petal is called tepal. So single units of perianth is called as tepals. Hope you got it. Now next one, a typical angiosperm's anther is bilobed, with each lobe having two theca, that is dithecous. So here if you are taking, you can see a typical angiosperm's anther. This is the structure of a typical anther. It will be having two lobes that will be connected to a connective tissue, and at the base you can see there is a filament also. Like this will be the structure of an anther. So there is a filament, and here you can see it will be also having anther, and both are getting connected to a connective tissue. This is the structure that is coming. So if you take the cross section of this, you can see it will be having almost like this in the cross section. So two lobes are coming in that. So like this, let this represent one lobe. In that you can see there are two sporangia. So like this it is bilobed and dithecous. So two lobes are coming, so it is bilobed with each lobe having two theca. Two thecas will be coming in each lobe and they are dithecous. The term that is given is dithecous. So generally the anther is dithecous. So the answer is A. Next question, often the theca is separated by, so here you can see the theca will be separated by longitudinal groove. A groove is coming, that is longitudinal. So the two thecas are actually getting separated by longitudinal groove. Now next question, the dithecus consists of how many microsporangia? In this given diagram you can see there will be 4 microsporangia. So dithecous anther will be having 4 microsporangia. So that is only given in the question. A dithecous anther consists of how many microsporangia? It will be having 4 microsporangia located at the corners and dash in each lobe, 2 in each lobe. So one lobe will be having 2 microsporangia and a complete anther will be having 4 microsporangia. Now next one, arrange the microsporangial wall in sequence of outside to inside. So if I take the cross section of an anther, the outermost layer that is coming in the anther will be a single layer, that is epidermis. Inner to the epidermis, you can see the next layer that will be coming is endothelium. So next layer, let me put it with the pink color. So this represents the endothelium, the second inner layer, that is also a single layer. Now inner to this endothelium, the next layer that will be coming, so this is having protection function, dehiscence also. Inner to that, the next layer that will be coming is the middle layer. Middle layer can be 1 to 3 layer of cells will be present. So that is the middle layer that is coming inner to the endothelium. Now inner to that, the next layer that can be coming will be tapetum. So like this, tapetum will be coming next inner to that. Inner to the tapetum only you can see there is sporogenous tissue. So sporogenous tissue will be present inner to the tapetum. Like that the sporogenous tissues also would be coming. Hope you got it about the sporogenous tissue. So these are the different layers in the anther. So what is the outermost layer? Epidermis. Inner to that endothelium. Inner to that middle layer. Then you can see there is the tapetum, the nutritive tissue. So the answer that we can take is B. Epidermis, endothelium, middle layer, tapetum. That is from outside to inside. Next one, how many of the microsporangial wall perform function of protection and helps in dehiscence of anther? The outer 3 layer, epidermis, endothelium, middle layer, they will be helping in protection, and the outer 2, endothelium, epidermis, if you take, dehiscence also is coming. Now next one, dash is responsible for nourishment of pollen grain, tapetum. Tapetum actually provide the enzymes as well as the hormones and the nutrients for microsporogenesis. So try to understand, there is sporogenous tissue that is present inside the microsporangia. So sporogenous tissue that is coming inside the microsporangia which is diploid, this will only turn into microspore mother cell that is also diploid. This microspore mother cell will be undergoing meiosis in order to form 4 haploid microspores. This is actually the microspore tetrad. This microspore tetrad is connected to each other by callose band, like this callose fibers are present. This has to be broken in order to break them into 4 microspores. Like this, you can see the microspore tetrad will be dissociated and they will be found in the single celled stage. They will be found in which stage? They will be found in the single celled stage. Then they will start undergoing unequal mitosis. They will start undergoing unequal mitosis and it will result in the formation of a big cell with an irregular nucleus that is actually called vegetative cell and the vegetative nucleus. In the cytoplasm of the vegetative cell, a small cell will be floating that is actually called as generative cell. So this is bicelled stage of a pollen grain. This is the bicelled pollen. In 60% of the angiosperm, you can see pollens are released in the bicelled stage, and remaining 40% you can see before the dehiscence they will be turning into generative cell, also will be dividing and they will be giving rise to 2 male gametes. This is the 3-celled pollens that is actually released in 40% of the angiosperm. So this is microsporogenesis. For this process to take place, you should understand the callase enzyme to break this microspore tetrad, or the hormones for this process to take place, even the nourishment of the pollens, etc., that is actually provided by the tapetum. So tapetum is the nutritive tissue, multinucleated, present inner to middle layer, keep on uptaking nutrients from middle layer and secreting it into the microsporangia. This is the main function of tapetum. Now next one, which of the following undergo meiotic division to form microspore tetrad? Just now I have discussed, that is the one that is given here we can take is only the sporogenous tissue. Actually it is the microspore mother cell that is directly formed from the sporogenous tissue. Generative cell mitosis only, microspore also mitosis only will take place. Next one, select the correct match. So already we have discussed this diagram. Outermost layer in the anther is epidermis. Inner to that, the next layer that is coming is endothecium. Inner to the endothecium you can see there is middle layer. So E represent middle layer, then B represent the sporogenous tissue or microspore mother cell, then A represent the tapetum. A represent the tapetum. So if you take the options in question number 13, you can see it will be B. Now next question. Microsporogenesis. Already we have discussed microsporogenesis. The formation of microspores or the pollens from the microspore mother cell. That is actually microsporogenesis. It's the process of formation of microspore which will later turn into the pollen grain from the pollen mother cell. It involves meiosis. So one meiosis will result in formation of 4 microspore which will further undergo 2 rounds of mitosis in order to form the matured pollen that is 3-celled, and in the 3-celled they will have 2 male gametes. So always remember, a pollen has 2 male gametes. Pollens represent male gametophyte that is producing the male gametes. The haploid structure that is producing the gamete, that is actually called as gametophytes. So male gametophyte is pollen in angiosperm. So all of these are actually correct. Next one, pollen grain represent, already I said it represent male gametophyte. So a matured pollen will be having a vegetative cell and its nucleus. Irregular nucleus will be present and its outer covering, that is the intine. Then external to that you can see there is a exine. At some location it is absent, that will be forming the germ pore. Now in the cytoplasm of this vegetative cell will be floating 2 male gametes. Like this, 2 male gametes will be also floating. Exine is made up of sporopollenin, intine is made up of cellulose and pectin. This much you might have learned. Next question, choose the correct things about pollen grains wall. Pollen grains wall, if you are taking, it has 2 layered prominent wall, that is correct. The hard outer layer prominent wall is exine, and exine is composed of sporopollenin, Then sporopollenin form continuous in exine. That is actually wrong. It's not continuous. At some location it is absent. That location where it is absent will be forming the germ pore. Through that germ pore only you can see the pollen tube will be emerging out. Hope you got this much. Now coming to the next question. Sporopollenin is absent in. Sporopollenin will not be present in intine. Intine will be having cellulose and pectin, and you can see the germ pore also it is absent. So both A and B. Next question. Pollen grains are well preserved as fossils because of presence of intine is wrong, presence of germ pores, presence of sporopollenin. Sporopollenin is a strong substance. It cannot be digested by any enzymes, acids or alkali. So for fossil forecast and all, this will be helping. Now next one, sporopollenin is degraded by. Sporopollenin cannot be degraded by any of the enzymes as well as the temperature, even acids or alkali. It is the most resistant organic material. So none of this can degrade the sporopollenin. Now next one, inner wall of pollen grain is called. Inner wall of pollen grain is intine. It is made up of cellulose and pectin actually. So it is thin, discontinuous. We cannot take any of this because it's not made up of lignin, it is made up of pectin. It's not discontinuous, it's actually continuous. So none of these options are matching for the inner wall of pollen grain. Anyway, inner wall of pollen grain is intine. Understand this intine is made up of cellulose and pectin. Next question. When pollen grain mature, it consists of two cells that are two male gametes only. So when the pollen grains are maturing, you can see there is a vegetative cell and there is a generative cell that is floating here. So bicelled stage can be observed. Then it consists of two cells that are generative and vegetative. So first statement is incorrect. It consists of two cells that are two male gamete is wrong. Actually it is consisting of two cells, that is generative cell and vegetative cell. Generative cell will be floating in the cytoplasm of vegetative cell. It consists of two cells that are male gamete arise from the vegetative cell is wrong. Two male gametes are actually arising from the generative cell. Sorry, that is actually wrong. Then it consists of three cells that are two male gametes developed meiotically from generative cell is also wrong. It is mitotically. Hope you got it. Now next question. Choose the incorrect statement among the following. In over 60% of angiosperm, pollen grains are shed at not 3-celled stage, 2-celled stage. So whenever they are reaching the 2-celled stage with one vegetative cell and generative cell, at that time itself they will be liberated in 60% angiosperm. So second statement is correct. Next one. Pollen allergy is not correlated with cause of Parthenium or carrot grass. They will be releasing the pollens in a huge amount. When it is inhaled, they can cause some infection in the respiratory pathway, making your immune system to produce allergic reaction. That means the mast cells will be producing histamine and serotonin that will produce an exaggerated response, that is called allergy. So carrot grass or Parthenium that can actually cause this immune system to produce what? Produce a higher response or allergy. So first option is correct. Then cause chronic respiratory disorders. There can be inflammation in the respiratory pathway, bronchitis and all can be observed. Carrot grass that come to India as a contaminant with not the rice, it actually came as a contaminant along with the wheat varieties. So this is actually a wrong statement. Then pollen grain of rice is viable up to 30 minutes. Understand, different plants have different pollen viability. In some plants you can see the pollens are viable only for a few minutes, like rice and wheat. It's only 30 minutes. In some of them you can see Rosaceae, Leguminosae, etc. You can see they may be viable for months and years, like that. So for a few months and all it may be viable in some plants like Leguminosae, Rosaceae, etc. But in some it can be years also. So viability of pollens will be varying in different plants. It may not be the same. So what is viability? The time period up to which they can retain the capacity of germination. That is called pollen viability. Now next one, which temperature is correct to store the semen for artificial insemination? So the semen as well as the pollen, all these can be preserved in liquid nitrogen at minus 196 degree Celsius for any number of days or any long time. So this is cryopreservation and all at minus 196 degrees Celsius. It's done in liquid nitrogen. You already know that, that is cryopreservation. So in pollen bank also this is actually done, and the pollens can be later taken for some breeding programs and all. Now next one, papaver show. So you should understand about the pistil or carpel. The female reproductive part of a plant is pistil or carpel, which will be having a swollen bottom, that is ovary elongated style, and there is a stigma. If there is only one carpel, that is said to be monocarpellary. Monocarpellary means, understand, there is only one carpel. If only one carpel is present, that is monocarpellary. Example, in mango, coconut, in all these you can see only one carpel is present. Whereas many carpels are present, that is called multicarpellary. Many carpels are present, that is called multicarpellary. In that itself, if the carpels are, so if you take a flower, in that, if the carpels are free from each other, many carpels are present, but each carpels will be free from each other without fusion, like this if they are standing, then it is called apocarpus. Then it is called apocarpus. Apocarpus means, understand, the carpels are free from each other. The condition is called apocarpus. Then if they are fusing, if they are fused with each other, if they are fusing with each other, then it is called syncarpus. So in NCERT some diagrams are given regarding apocarpus, syncarpus, etc. So here papaver that is given in NCERT, it is an example of syncarpus and multicarpellary. Papaver. So many other example if you want I can give. Apocarpus example, lotus, rose, Michelia, all these are example of apocarpus, whereas papaver given in NCERT as well as Solanaceae family that you are learning in morphology of flowering plants, it's axile placentation where it is multicarpellary. Then Liliaceae you can see, even the mustard family, Brassicaceae, all these are multicarpellary. And syncarpus where the carpels are fused together. So connection questions also when you are doing for NEET, you can see some connection question with morphology chapter. So this outline you keep it and try to connect with the morphology of flowering plants also. So understand, if only one carpel is present, that is monocarpellary. Example from morphology chapter, mango, coconut, superior as well as monocarpellary ovary is coming in mango and coconut. Then the pistil or carpel, if many are present, that is called as multicarpellary. Here it is multicarpellary. Many carpels are coming. If they are free, it is called apocarpus. If all of them are fusing together, that is called syncarpus. So remaining all we will discuss in morphology chapter, that connections and all I will be telling. Now next one. So the answer here is B. Given diagram is of. Given diagram you can see many carpels are present and each carpel is alone. They are not fusing with each other. This diagram is multicarpellary and apocarpus gynoecium that is given in NCERT and it is of Michelia. So the answer is 1. Next one, which of the following serve as a landing platform for the pollen grain? So you know that what is pollination? Transfer of pollen grains from anther to stigma of a flower is called pollination, and the landing platform of a pollen grain is stigma. So stigma is the landing platform. So page number 25, this question is there. The next one, choose the correct statement. Inside the ovary is the ovarian cavity, also called lodicule. It's actually locule. It's also called locule. Then megasporangia is commonly called as ovules. That is the correct statement. So it is called locule, and here in the ovarian cavity there is placenta. To that only ovules are attaching. Then megasporangia is commonly called as ovule. That is correct. Another name for ovule is megasporangium. Then placenta is located outside the ovarian cavity is wrong. Actually it is located inside the ovarian cavity. So only B statement is correct. Now choose the incorrect statement. Number of ovules in an ovary is 1 in case of paddy. That is correct only. So some plants have only one ovule. Mango, paddy, rice, all these if you are taking only one ovule per ovary is coming. Then number of ovule in an ovary is many in papaya. Just try to understand how many seeds are coming. So number of seed means number of ovule, because ovule only become the seed. Papaya many seeds are there. So understand they have many ovules. The number of ovules in an ovary is 1 in orchid. Actually, orchid have many small seeds. So they should have many small ovules also. That statement is actually going wrong. Then week 10 mango, only one ovule is seen. So from this, hope you got the example of plants which are having only one ovule per ovary and many ovules also. So the fruits which have one seed and many seed, like that you take. One seed, one ovule, many seed, many ovules. Next one, ovule is attached to the placenta with the help of a stalk-like structure only they are attaching. That is called funicle. Now let me tell the structure of a funicle. So inside the flower, you can see there is ovary. So inside the ovary, they are attaching with the ovule with the help of a stalk-like portion. The stalk-like portion that is coming here is called as funicle. Then the point of attachment is called as hilum. Then the base portion is called chalaza. The base portion of the ovule is called chalaza. Then from that you can see some coverings will be coming out. From the chalaza arises the covering of the ovule. That covering of the ovule is what? The covering of the ovule is integuments. The covering of the ovule is called as integument. So there is an outer integument and there is a inner integument. Like this, integuments are coming. At a point they are not fusing with each other. That region is called as micropyle. So if you are taking the structure of an ovule, you can see there is a funicle, the stalk portion of the ovule. The point of attachment is called hilum, and you can see there is a chalaza. From the chalaza arising is the integument, outer integument, inner integument, and the point of where they are not fusing with each other is called as micropyle. Inner to the integuments, you can see there is nucellus. Inner to the integuments, there is nucellus also that is coming. Like this, nucellus will be coming inner to the integuments. This nucellus is the nutritive layer. They will be giving the nutrients to the developing embryo sac and all. So inner to that only the embryo sac will be coming with 3 antipodal, polar nucleus, and the egg apparatus with egg. So this is the structure of a typical ovule, where you can see it is 180-degree inverted like this. This type of ovule is actually called anatropous ovule. This type of ovule is called as anatropous ovule. So this is funicle. Now next question. Hilum represent the junction between ovule and the funicle. So it is the junction between ovule and the funicle. That is hilum. Now next one. Chalaza end represent. Chalaza end will be representing basal part of the ovule, which is attaching to the funicle. Then next one. Female gametophyte of angiosperm is represented by embryo sac. Embryo sac is the female gametophyte. How it is developing and all I will tell later. Then next one. An ovule generally has dash embryo sac formed from a megaspore through dash division. So an ovule will be generally having a single embryo sac. It will be having a single embryo sac that is formed from a megaspore through megaspore through mitotic division. It will be through mitotic division. So here it is single and equational. Next question. Megasporogenesis is not related to. Megasporogenesis is the process by which a embryo sac is formed from megaspore mother cell. So you know that a cell inner to the nucellus will be turning into megaspore mother cell. So one of the cell inner to the nucellus will be turning into megaspore mother cell. This megaspore mother cell, which is diploid, can undergo meiosis, and it will be resulting in the formation of 4 megaspore. In that 4 megaspore, 3 of them facing the micropylar end will degenerate. 3 of them facing the micropylar end will degenerate. One which is facing the chalaza, they will become functional megaspore. This functional megaspore can undergo repeated nuclear division. First round of division, it will form 2 nucleus, and in that you can see one move to the chalaza, one move to the micropyle, and after that they will undergo second round of division that will result in 2 nucleus at chalaza, 2 nucleus at micropyle. Then they will undergo third round of division. That third round of division will result in the formation of 8 nucleus. After this 8 nucleus formation, 2 from each pole move towards the center. So like this, there is 3 at the chalaza, 3 at the micropyle, 2 at the center. Now you can see they will be undergoing cytokinesis. In cytokinesis, when occurring, 3 at the chalaza will be resulting in the formation of antipodals, whereas 3 at the micropylar end, in that 2 of them will be turning into synergids, which is having filiform apparatus, and one will turn into the egg. This egg and the synergid together is called as egg apparatus. This is actually called as egg apparatus. Then this large cell at the center is called central cell, which is having 2 nucleus coming from each pole. That is actually called as polar nucleus. So like this, they will be resulting in the formation of 7-celled 8-nucleated embryo sac. So there is a 7-celled 8-nucleated embryo sac. This was first reported by Strasburger in Polygonum plants. Hence it is called Polygonum type embryo sac. It is called Polygonum type embryo sac. Hope you understood the Polygonum type embryo sac, 7-celled and 8-nucleated embryo sac. So here the formation of this embryo sac is from a single functional megaspore. So that we can call it as. So like this formation is from a single functional megaspore is turning into this much. This is called monosporic development. This type of development is called as monosporic development. Development of a functional embryo sac that is coming from the single functional megaspore, that is monosporic development. Now let's look at the option formation of megaspore from megaspore mother cell. That is correct only. From a megaspore mother cell, by repeated mitotic division, 3 round of mitotic division, they are forming it. So that is correct. So not related to megaspore mother cell. So formation of megaspore from megaspore mother cell, that is by meiotic division. Then megaspore mother cell undergo meiotic division for megaspore formation. So that is also correct. Megaspore mother cell is undergoing reduction division to form megaspore. Then formation of microspore. Anyway, this is not coming here. This is megasporogenesis. You don't have to look at microspore. Now next one. Ovules generally differentiate as a single megaspore mother cell in micropylar end. Understand? The answer is this one. Understand? Inside the ovule, there is funicle, there is integument, chalaza, etc. So here just imagine there is nucellus also, like this nucellus is coming. So one of the cell that is coming at this end, is turning into the megaspore mother cell. They will undergo division to form 4 megaspore. In that, you should understand 3 that is located towards the micropyle will degenerate. This 3 will degenerate and 1 will become functional, which is facing towards chalasa. So ovules generally differentiate as a single megaspore mother cell at the micropylar end. At this location only this is happening, and this one will actually enlarge and they will be turning into the embryo sac with polar nucleus, antipodal and the egg apparatus. This is what is happening. So all the things are located towards the micropylar end. Next question. In a majority of flowering plants, one of the megaspore is functional while the 3 will degenerate. That is correct only. All 4 megaspore can develop into a female gametophyte. No. Embryo sac is actually formed from a single megaspore. That is actually called as, what is it called? Monosporic development. So megaspore is actually formed, embryo sac is formed from a single functional megaspore. That is called monosporic development. Then 3 megaspore is functional while the other one degenerate. That is also wrong. Only 1 megaspore is functional and the remaining 3 will degenerate. That is what we call it as monosporic development that we were discussing at this one. So a megaspore mother cell, meiosis, 4 megaspore, 3 degenerate, 1 will become functional. That is monosporic development. Next one, monosporic embryo development involved 1 functional megaspore, 1 haploid cell formed in egg apparatus. That is wrong. 4 functional megaspores. So the answer is 1 only. Next one, choose correct statement. The nucleus of functional megaspore divides mitotically to form 2 nuclei which move to opposite pole, forming 2 nucleated embryo sac. 2 more sequential mitotic nuclear division in 2 nucleated embryo sac result in the formation of 4 nucleated. 2 more sequential mitotic nucleus, so 4 nucleated. Mitotic division in embryo sac formation up to 8 cell is strictly free nuclear. So all of these statements are correct. First, it will be having a single nucleus. Then first round of division will result in 2 nucleus. In that they will move towards opposite pole. There they will undergo the second round of nuclear division in order to form 4 nucleus, and this will continue until it's becoming 8 nucleated embryo sac. Then after that only cytokinesis will take place. In that this 2 will move towards the center like this. So 3 at here, 2 at the center, 3 at here. Then cytokinesis will be resulting in the formation of 3 antipodal that is coming at this location. Then you can see there is a central cell with polar nucleus. Then there is synergid with the egg. Like this 7-celled 8 nucleated embryo sac will be produced. So all the statements are correct. Next one, how many of 8 nuclei of typical embryo sac is surrounded by cell wall? You can see 6 of them. Central cell is not actually having a pure cell wall. So antipodals, the 3, synergids and the egg, that is properly surrounded by the cell wall. So 6 are actually surrounded by the cell wall. So here the 8 nucleated embryo sac, if you are taking the polar nucleus containing central cell, is not actually having a cell wall. So remaining 6 antipodals as well as egg and the synergid, it is getting surrounded by the cell wall. Now next one, central cell of a typical embryo sac is situated below the egg apparatus. It is actually located below the egg apparatus. Then next one, choose the correct statements about egg apparatus of a typical embryo sac. It's situated at the micropylar end. So if you are taking the inverted ovule, an atropous ovule, you can see in the embryo sac, the egg apparatus is coming near the micropylar end with 2 synergid and 1 egg. And here is the polar nucleus. At the chalasal end is the antipodals. Then it consists of 3 cells, 2 synergid and 1 egg. So both A and B are correct. Next, a typical embryo sac of angiosperm at maturity is 7-celled and 8 nucleated. That only we have discussed. In that 7-cell, 3 are antipodal, 3 are forming egg apparatus, that is having an egg with 2 synergids and filiform apparatus here, and 1 central cell with 2 polar nucleus. So it is 7-cell but having 8 nucleus. In that, central cell is dual nucleated. This is the most common embryo sac in angiosperm, that is polygonum type embryo sac, which represent the female gametophyte. Next one, select the correct match. A represent antipodals, A represent antipodal, B represent polar nucleus, B represent polar nucleus, C is the central cell, C represents central cell, D represent egg, E represents synergids, and F represent the filiform apparatus. F represent the filiform apparatus. So here A is the answer. Next one, pollination is, it is the transfer of pollen grains from anther to stigma of a flower, that is pollination. So it is the transfer of motile pollens. Pollens are actually not motile. Then transfer of non-motile anther, then transfer of non-motile pollen to the stigma, that is actually called pollination. Next one, transfer of pollens from anther to stigma of same flower is called as, it's called as autogamy. So if I take a flower, in that flower, if the pollens are actually moving from anther to stigma of the same flower, so like this, in the flower, if you can see, there is movement of pollens from anther to stigma of the same flower in the plant. Like this it is moving. This is actually self-pollination. It is autogamy. This is called as autogamy. So what is autogamy? It is genetically and ecologically self-pollination. Genetically and ecologically, autogamy is called as self-pollination. Whereas, if the pollens are moving from anther to stigma of different flowers in the same plant, then it is called as geitonogamy. Then the process is called geitonogamy. So you should understand, if the pollens are actually moving from anther to stigma of different flowers but in the same plant itself, then it is called, so like this if it is moving, then it is called as geitonogamy. Geitonogamy, genetically it is self-pollination and ecologically it is cross-pollination. Then if the pollens are moving from anther to stigma of different flowers in different plants but of the same species, like that a flower is there from the anther to stigma, if the pollens are moving, then it is called synogamy or allogamy, that is genetically and ecologically is considered as cross-pollination. So like this it is moving, that is actually synogamy. Hope you got it. Sometimes diagrammatic questions also will be coming, so you should be able to identify this. You can use this diagram. Now next question. Read the given statement. Autogamy cannot occur in open flowers. That is actually incorrect. Autogamy is possible in open flowers if the pollen release as well as the pistil's maturity is occurring at the same time. There is chance of self-pollination. So statement 1 is incorrect. It can occur in open flowers if you can see the pollens as well as the pistil, or the stamen and pistil is maturing at the same time, and if there is no self-incompatibility, there is chance of self-pollination. So statement 1 is wrong. Geitonogamy cannot occur in closed flower. If the flower is closed, there is no chance of geitonogamy. They can undergo autogamy, but they cannot transfer the pollen between the flowers. So that is actually correct. So statement 1 is wrong, but 2 is correct. Next one. Complete autogamy is rare in. So autogamy, if you are taking, you can see this will be very rare in case of open flowers. Open flowers generally they will be undergoing which pollination? Geitonogamy or synogamy. There is more chance. So it will be more occurring in closed type of flower and it will be very rare in open flowers and all. Now next one, flowers that do not open at all, that is called as cleistogamous flower. Always the flowers are closed and these flowers are invariably autogamous. So if a flower is not at all opening, then that is called cleistogamous. Cleistogamous flowers are invariably autogamous. They can undergo only self-pollination. Whereas chasmogamous is open flowers. So there are some examples given in NCERT which possess both the type of flower, Commelina, Viola, Oxalis. These are actually having both open as well as closed type of flowers. Now next one, Oxalis. Oxalis, Viola, Commelina, they are having both cleistogamous and chasmogamous flowers. Now how many of the given characters are necessarily present in cleistogamous flower? Cleistogamous means they are actually closed flowers. Anther and stigma lie close to each other. So cleistogamous flowers mean surely they should undergo self-pollination. So adaptations of self-pollination will be seen in closed type of flowers, cleistogamous flowers, where you can see anther and stigma will be lying very close to each other and there is synchrony in pollen release and stigma receptivity. Release of pollen grains and acceptance of pollen by the stigma, you can see it will be synchronized. It will be taking place at the same time. So that is correct only. 1 and 2 are correct. Length of anther and stigma are very different is wrong. Maximum the length of the filament as well as the length of the style in the pistil will be almost same, so that there is more chance of self-pollination. They won't show heterostyly. Flowers is necessarily dioecious. It's not dioecious. Sexes won't be separate if it is cleistogamous. They will try to have a bisexual flower. Dioecious means what? Sexes are separate. They will be monoecious. You can see they will be having both the stamen and pistil in the same flower. They will be monoecious only. Then assured seed set even without pollinators. They don't need any pollinators. So here you can see given 3 statements are actually correct regarding cleistogamous flowers. Now next one, gytinogamy is, you know what is gytinogamy? Transfer of pollen grains from anther to stigma of different flowers but present in the same plant. It's ecologically cross-pollination but genetically it is self-pollination. So genetically self-pollination, functionally or ecologically it is cross-pollination coming to gytinogamy. Now next question. Cynogamy, the transfer of pollen grains from anther to stigma of different flowers in different plants but of the same species. Genetically, functionally or ecologically, it is cross-pollination. So functionally self-pollination wrong, genetically cross-pollination both genetically and functionally. Then autogamy. Autogamy, you can see it is self-pollination in the same flower itself. So it is self-pollination both genetically and functionally. Then genetically different type of pollen is brought to stigma in cynogamy. In cynogamy you can see genetically different will be coming. Autogamy, gytinogamy, genetically it is same, but cynogamy it is genetically different. Now next one, pollination by abiotic agent is a chance factor, that is correct. Pollen is produced in enormous amount as compared to number of ovules. That is also correct. Both A and B are actually correct. So I can tell B is the reason for A also. Because since it is a chance factor, they will try to increase the possibility of the pollens falling on the stigma of the plant. For example, if it is carried by the wind and all, they will produce enormous pollens. Because it's a chance only. Sometimes only they may fall on the stigma. So to increase the possibility, enormous amount of the pollens are produced and all. So here, answer to the question, A and B are correct and B is the reason for A. So both are actually correct only. Hope you got it. Next question. Which is the more common abiotic agent of pollination? You know, there are some agents of pollination, abiotic and biotic agents of pollination. Abiotic means the non-living. So here wind and water is the most common abiotic agents of pollination. In that itself the most common is wind pollination. Pollination by wind is also called anemophily. Then insect and animals, they are also pollinating. They are biotic agents of pollination, and in insect is the most common biotic agent of pollination. In that itself, bee pollination is more common. That is entomophily only, you can see. Then next question, the pollen grains in wind pollinated plants should be. Wind pollinated plants, you can see, they produce enormous amount of pollens. That is lightweight, non-sticky, and they will be releasing a lot of pollens into the wind. And this wind will be carrying them to the female flower, like corncob and all. There is tassels in the corncob that will be fluttering in the wind. That feathery stigma will be trapping these pollens for pollination. So this is how the pollination is occurring by the wind. So here it will be lightweight and non-sticky, produced in enormous amount, and they have a feathery stigma. So these are the adaptations of plants that are pollinated by wind. So example, grasses, corncob, etc. Then next one, wind pollinated flowers often have dash in each ovary. How many ovules in each ovary? And flowers are often dash. You can see majority will have a single ovule in each ovary, and they are packed into inflorescence or clusters. Next one, tassels in corncobes they are actually stigma and the style. There will be a feathery stigma that will be fluttering in the wind to catch the pollen grains that are coming out. Next one, match the columns 1 with column 2. Wind pollination, maize and all, you can see it is wind pollination. Water pollination, that is observed in monocots. So you might have read the textbook line, the pollination by water, fresh water, it is hydrilla. So it is 4B. Then water pollination, majority monocots. In angiosperm aquatic plants, if you are taking, there are 30 genera. Mostly they are monocots only. Even algae, bryophyte, pteridophyte need water for completing life cycle. So hydrophily, if you are taking in angiosperm, very less aquatic plants are there. In that, majority are monocots, and it includes 30 genera. So that is water pollination or hydrophily. So it is 2C. Then biotic, amorphophallus. You know about amorphophallus, 6 feet height plant that is pollinated by the birds. Ornithophily is occurring in them. ACDB, ACDB. Now next one, distribution of some bryophytes and pteridophyte is limited. Transport of male gamete in bryophyte and pteridophyte is dependent on water. So plant kingdom chapter you might have learned. For completion of life cycle, both bryophyte and pteridophyte depends on water. The first land plant is bryophyte, but they need water for completing the life cycle. Anyway, they are found in the watery environment. But coming to pteridophyte, they are the first true land plants, but they were unsuccessful because they also require water for completing the life cycle. In order to transfer the male gamete to the female gamete, they also require water. That is the reason they became unsuccessful. So here, both bryophytes and pteridophyte, transport of male gametophyte from their antheridia to the archegonia needs water. So you can see both the assertion and reason are correct, and reason is the correct explanation of assertion. Next one, aquatic plants pollinated by water are given except Zostera, marine seagrass, that is actually pollinated by water only. Then hydrilla, found in fresh water, that is also hydrophily. Water hyacinth and water lily, though it is found in water, their flowers will be emerging above the surface of water, and it is pollinated by wind or insect. So it is not pollinated by water. Now pollination in water lily occurs by wind or insect, because I already said water hyacinth and water lily, the flowers are not actually seen inside the water, it's coming above the surface of water, and it is getting pollinated by wind or insect. Next one, choose the correct statement for pollination in sea grasses. Sea grasses, female flowers reach the surface of water. Female flowers remain submerged. That is correct. in sea grasses, Zostera, you can see it is hypohydrophily. The male and female flowers are under the water only and pollination is inside the water. Then pollens are released on the surface of water is wrong. Actually, pollens are released inside the water and it is a long ribbon-like pollens that will be released. The longest pollens is in Zostera. Then pollen grains are carried passively by the water. Under the water current will be carrying them. So that is also correct. So it's not actively, they don't need any energy and all. Then most of the pollens reach stigma. Majority will not be reaching. Some of the pollens only will be reaching the stigma. So here 2, 4, 5 and 8 are the correct statement. So sea grasses or Zostera, you can see they have a long ribbon-like pollen that is covered by a mucilage in order to prevent wetting and all, and they will be releasing the pollens under the water. Both male and female flowers are underwater only. From the male flower, the pollen that is released that will be passing through the water current and will be entering into the female flower. So it is hypohydrophily. Then in case of Vallisneria, it is epihydrophily. The pollination is occurring above the surface of water only. Female flowers are above the surface of water, male flowers are underwater. Pollens release will be swimming and reaching on the surface. So here the female flowers remain on the surface of water, pollens released on the surface of water. All these points are related to Vallisneria. But whatever I discussed regarding this D option, that is coming for sea grasses. Okay, next question. Choose the correct statement for pollination in Vallisneria. So already we have discussed about the pollination in sea grass, that is hypohydrophily, where you can see the pollination is occurring under the water. But coming to Vallisneria, it is occurring above the surface of water. So female flowers reach the surface of water, so it's not submerged. Then pollens released on the water surface. Pollens are actually released on the water surface, not inside the water. Then pollen grains are carried passively by the water. So slowly they will be floating to the surface. So you should understand the male flowers are underwater and female flowers will be on the surface. So the pollens that are released will be coming above the surface of water and some of them will be entering into the female flowers. So it is carried passively only, not actively, and you can see only some of the pollens will be reaching the stigma. So it is 1, 3, 5 and 8. 3, 5, 8. Okay, that's all. Next one, requirement of pollen of water pollinated plants are the pollens should be light. Then pollens with mucilaginous covering in order to prevent it from wetting. Non-sticky pollens, we can't tell it is non-sticky or sticky. Then long ribbon-like, that long ribbon-like and all is seen in Zostera only. That ribbon-like pollens, not all the hydrophily, you cannot see this ribbon-like pollen, that is only in Zostera. So the answer is both 1 and 2. Next one, majority of angiosperm use dash for pollination. Majority angiosperm, you can see animals are the pollinating agent. They will be putting some floral rewards in order to attract the pollinators. So it is biotic agents only. Next one, identify the given labels. What is 1, what is 2? 1 is closed flowers and 2 is open flowers. So what is closed flowers? Cleistogamy, where you can see it is autogamous, and open flower is chasmogamous, where you can see allogamy. Allogamy will be most common. So given option, the most apt is C only. Then next one. So example of cleistogamous and chasmogamous, which is common in Commelina, oxalis, viola. Don't forget that also. Next question. Dominant biotic pollinating agents are pollination by bee. You should also remember the characters of flowers that are pollinated by the honeybee. The floral rewards include nectar. Nectar will be provided and even the pollens and a good fragrant flower. Even the pollens will be sticky, yellow in color. These all things will attract the bee pollinators. Most common biotic agent of pollination in animal is bee pollination only. Next one, how many of the following may act as pollinators? Bees, butterfly, wasp, beetle, bats. This much will be acting as pollinators. Next one, insect pollinated flowers are. You can see they may be large, colorful, rich in nectar. That is correct. Large, colorless, we can't tell, it is colorful only. Then it can be small, clustered, fragrant, sticky. So these two are actually matching. So more than one option is correct. Floral rewards are anything that is provided by the flower in order to attract the pollinating agent is called as floral rewards. So here nectar, pollen grains, all these are actually the floral rewards in order to attract the pollinators. For example, bee pollination, these two are the floral rewards.